Well, good morning. Thank you all for joining us today. We're really excited for today's session. My name is Terrence Henry. I'm the Executive Director of TAMEST. That stands for the Academy of Medicine, Engineering, and Science of Texas. And we're really excited today to have this research presentation session from our recipient of the Edith and Peter O'Donnell Award in Medicine for 2021. Before we begin, I'd like to tell you a little bit about our organization. TAMEST supports collaboration to advance research, innovation, and human talent development in the state of Texas. We are composed of more than 320 Texas-based members of the 3 national academies. So the National Academy of Medicine, National Academy of Engineering, and National Academy of Sciences, as well as our state's 11 Nobel laureates. We're supported by the founders of our endowment, as you can see listed here on this slide, as well as our 18 academic and medical research member institutions. We want to thank all of them for their support in helping make these awards possible, as well as their ongoing contributions to the research community in our state. So as I mentioned, today is our first session highlighting the research of this year's recipients of the Edith and Peter O'Donnell Awards. We're going to be having 4 of these sessions in all over the coming weeks. And we're really proud to present these researchers and give them an opportunity to showcase their work, because these recipients really play a vital role of helping us recognize and promote the rising star researchers in our state. These awards are named in honor of Edith and Peter O'Donnell, who have been steadfast supporters of TAMEST since our inception and incredible advocates for excellence in education and research in Texas. The awards are supported through the Edith and Peter O'Donnell Endowment, and we'd like to thank them and acknowledge their contributions, as well as the O'Donnell Foundation for their generous contributions to the awards program. A little bit about how the awards are selected. We have a selection committee, which is composed of 12 TAMEST members serving on 4 subcommittees. There's 1 for each of the awards. And we'd like to recognize our 2021 awards committee, and that was chaired by Kenneth Arnold. And they did a lot of hard work evaluating these highly qualified candidates. So we'd like to thank them and recognize them. We'd also like to thank all of the Nobel laureates who serve on the Nobel Laureates Review Committee for reviewing the finalists for the awards. Now to today's session. This is going to be a 20-minute research presentation from the recipient of the Medicine Award, Dr. Benjamin Arenkiel. After he concludes his presentation, we'll have some time for audience questions. And you can ask your questions using the Q&A function in Zoom. It's a box you click at the bottom of your Zoom screen. And you can ask your question at any time during the presentation or during the Q&A segment itself. Now, I'm very excited to introduce our moderator for today's session, Dr. Malcolm Brenner. Dr. Brenner is the Fayez Sarofim Distinguished Service Professor, Founding Director, Center for Cell and Gene Therapy, and Professor, Departments of Medicine, Pediatrics, and Molecular and Human Genetics at Baylor College of Medicine. Dr. Brenner is a TAMEST member and also served on the 2021 O'Donnell Awards Committee as the Medicine Subcommittee Chair. Dr. Brenner, thank you very much for your service to TAMEST and for moderating today's session. We'll turn it over to you.
Thanks, Terrence, and thanks to the TAMEST team for organizing this session. So it's a very great pleasure to introduce Dr. Benjamin Arenkiel. He's a colleague from my own institution, Baylor College of Medicine, and he's currently an associate professor in the Department of Molecular and Human Genetics and a member of the Jan and Dan Duncan Neurological Research Institute. Dr. Arenkiel obtained his doctoral degree in genetics at the University of Utah, where he worked with Mario Capecchi. He was investigating the genetic mechanisms of brain development and then went on for his postdoc to Duke University. He worked initially with Larry Katz, and after his untimely death, he joined Michael Ehlers, where he investigated the architecture and the signaling properties of brain circuitry. And even as a postdoc, he published very extensively and won several awards. So we were very delighted when in 2010, Dr. Arenkiel joined the faculty of Baylor College, and he was in fact our very first McNair Scholar. He's been working on how genes and sensory experience interact to build, maintain, and modify neural connections in the adult brain. and can thereby produce long-term influences on behavior. This work has already won several awards and he was selected for the O'Donnell Award in Medicine because he's now identified specific circuits that combine sensory perception and motivational states and can thereby influence addictive and aversive behaviors like eating disorders and perhaps other neuropsychiatric diseases as well. And this work provides a future opportunity for entirely new therapeutic interventions. So please join me in welcoming Dr. Benjamin Arenkiel, winner of the 2021 O'Donnell Award in Medicine.
Okay, thank you very much, Malcolm, for the introduction. Let me swap my screen over real quick. Oh. Okay, is this up? Okay, thank you very much, Malcolm, for the thoughtful introduction. And also, I must for sure thank the Edith and Peter O'Donnell Foundation. This is really a fantastic mechanism, and it really supports Texas advancement of science, and it's remarkable across the board. In addition, I also have to, of course, thank TAMEST for their efforts in recognition, efforts in selecting people in the recognition of this prestigious award. So without any delay, I'm going to give you a general overview and introduce you to my teams, to my research that we're doing in the laboratory. Overall, we have kind of an ambitious goal of understanding how the brain is built and how it actually works. This starts from the level of, swap over to the laser pointer, starts at the level of genetic codes and how it differentiates and specifies different neurons in the nervous system, how these neurons then go to form given connections, then ultimately how these connections form what we think of as complex neural circuits that govern overt physiologies and behaviors. And in fact, this is kind of what we've advanced to recently, is understanding how these connections really guide all features of our behavior and physiology. Why do we study the basic features of circuit architecture and function? Is that really, when it boils down to it, I think it's pretty well accepted that all of the bad things that go wrong with brain tissue really evolves from dysfunctional brain circuitries, ranging from neurodevelopmental disorders to addictive and neuropsychological behaviors. So we're faced with a significant challenge, and at face value, the challenge we're up against is really a numbers game. It's pretty well appreciated that there's over 1 billion neurons that make up the brain, and these neurons themselves can have up to 1 trillion different connections. And in fact, this has actually been a speaking point to much of popular science lately. If you've looked around the typical journals and documentary type shows, is that we've have quotes such as the most complicated organizational matter we know, most complicated object in the known universe, the most complex mass of protoplasm in the galaxy. If we stretch out the brain, it can reach to the moon and back. And probably my favorite is that it's the next big frontier. I tend to agree with these comments, but obviously they're sensationalized a bit. Usually when I like to describe this, in particular to Texans, I use a little bit more tractable analogy and a scale image where you can see this is what 1 trillion pennies would be to a typical football field. So elucidating 1 trillion different patterns of connections is really a big goal if we speak about this glibly. However, I'm going to kind of give you the punchline of what we're working on, and what I'm going to try to lead up to in the end is that I think that uncovering the complexities of these circuits in looking at them in a little bit simpler way is that if we can elucidate basic circuit architecture, circuits that are overlapping or redundant, and actually exploiting this perceived complexity might be a novel avenue to actually treat dysfunction and disease of the nervous system. But to start somewhere, especially in a basic laboratory, we embrace the mouse model as our favorite model of choice. And that's basically because we can genetically engineer it at will, cells, circuits, entire brain regions. And really, we're only limited now by our own creativity and how we genetically engineer the mouse. And in fact, the analytical tools have advanced significantly over the years that allow us really to interrogate brain circuits, map and manipulate their function to understand what they're doing. We also embrace a model within the model. So shown here is a pseudo-colored image of the mouse olfactory system. This is kind of what we reduced and initiated a lot of our studies with. And the reason why we like the mouse olfactory system is multifold. First of all, it's directly related to almost all of mouse behavior. It's a great input-output relationship that we can assay at the level of the model, where we don't have to necessarily think about the human. It's experimentally tractable. Its anatomy is fairly well characterized, and as far as the sensory system goes, it's relatively simple. It bypasses a lot of the complicated relay stations of the thalamus. But interestingly, it's endowed with this unique feature of ongoing adult neurogenesis. This also occurs in the hippocampus, but really what it allows us to have is a reservoir of cells that are continuously being generated and integrating into adult brain circuits on a daily basis. And really a big focus of my laboratory still to date has been to try to elucidate the mechanisms of how these new neurons are continually wiring up in hopes of kind of co-opting or pirating some of these mechanisms to use it in other parts of the brain to repair and replace damaged or diseased nervous tissue. So this has been a large focus of our laboratory, and it's really been the seeding ground for what we're doing right now. Along the way, details I won't really go into, we identified one particular signaling mechanism that we became intrigued with. And this is the cholinergic basal forebrain signaling mechanism. This is shown in illustration form here, where the cholinergic basal forebrain resides deep in the brain and sends long-range projections to the olfactory bulb and makes selective connections onto adult-born neurons. And we became intrigued with this as a candidate to govern brain plasticity and adult-born neuron wiring for a handful of reasons, largely because it synthesizes the neurotransmitter acetylcholine. Acetylcholine caught our eye for a handful of reasons. First of all, it's known to influence components of olfaction. It's associated with the olfactory system. It's synaptogenic in vitro. It's been linked to reward, arousal, plasticity. And interestingly, studies looking at Alzheimer's models treated with acetylcholinesterase blockers, which degrades acetylcholine, or show increased adult-born neuron integration. So this was a really nice candidate for maybe acetylcholine is directly guiding the integration of these adult-born neurons. So to address this, we took our typical genetic approach in mouse. And what I'm going to be doing for a lot of these data slides, since we're talking to a broad audience, is I'm going to kind of show this in illustrated, larger concept-wise. And then those that are interested can look at the alleles or the different viral manipulations if you want more detail, and I can clearly address this at the end if you're interested. But sticking to the larger concepts, what we basically did is we went into the cholinergic basal forebrain and selectively ablated the cholinergic neuron, sending projections into the olfactory bulb using a diphtheria toxin method. Now, this is where our whole research program kind of has taken a tangential turn into the real world of serendipity in finding novel brain circuits that are associated with the title of my talk, associated with reward, plasticity, feeding disorders, and neuropsychiatric disease. And because when we knocked these cells out, to our surprise, we generated these morbidly obese mice. Now you can see here a control mouse is roughly 25 grams. These mice rapidly gain weight to the point of almost triple the weight. And in fact, if we let them go, they can get up to 80 grams. What we noticed is that this is correlated to this body weight increase happens very, very quickly over a period of just a couple of months. And it's directly correlated with food intake. Through exhaustive analysis that I'm not going to show you, we also basically found out that this is not a metabolic dysfunction. This doesn't arise from some diabetic model. If we assay serum hormones, serum factors, as well as all of the different type of respiration components, we found out that this is basically a behavioral problem. This is a problem of the brain. In short, what we learned is that this basal forebrain controls appetite in a way that creates an insatiable appetite. When we knock these cells out, you have unchecked food consumption and a bona fide hyperphagia. So this is how we went from neural stem cells, synapses, and plasticity to basal forebrain circuits governing feeding, addiction, and aversion. And this is kind of what I'm going to be focusing on now. But just as kind of an interesting highlight is that I think ultimately this notion of these different types of physiological states are not very far off from the same mechanisms that are gating plasticity. So as we all know, that many of the hypothalamic-driven behaviors such as feeding, sleep, stress, thermoregulation, this all is borne out through tight control of homeostatic mechanisms between the periphery and a region in the brain called the hypothalamus. this has gained a lot of attention, and in particular towards feeding. We completely appreciate as a field now that much of feeding is governed by the brain. However, not much attention has been given to the non-homeostatic mechanisms that gate the homeostatic mechanisms. Anecdotally, we all know that things like mood, sensory cues, and what we think is being rewarding or aversive directly impinge upon things like sleep, eating, stress. And interestingly, along the way, what we think that we had found serendipitously is that the cholinergic basal forebrain, for its known role in motivation, sensory processing, reward, and aversion, might be the missing link that are coupling these non-homeostatic mechanisms with the homeostatic mechanisms. So we decided to kind of dig our heels in on this particular research angle and try to figure out what's actually going on here. So the first approach we took is we kind of backpedaled and took a similar genetic approach of, in this case, not destroy or killing or ablating or shutting the cells down, but activating them. In this way, we targeted the same population of cells in the cholinergic basal forebrain, but this time we expressed a factor called NACBAC. It's basically a sodium channel from bacteria, and it's leaky. So what it does is any cell that it's expressed in, it basically elevates their activation pattern and makes them fire more. Consistent to what we had seen before, the mice that had activated cholinergic basal forebrain circuits rapidly lost weight and became severely gaunt and lost weight at a rate that was borderline anorexia. This was coupled to dramatic decrease in food intake, and notably this was voluntarily. And body weight precipitously dropped over a period of days. In fact, if we didn't intervene within 2 weeks, these animals would actually starve themselves to death. Now this at least deserves a little bit of highlight because it's really hard to, it's pretty easy to get a mouse to become obese, but to actually make an anorexic mouse and have a mouse stop eating is pretty remarkable. It goes about against everything evolutionarily that they've been programmed to do. And in fact, we validated that this was bona fide hypophagia because we could do the same type of thing, look at all the serum hormones, and these were all normal until, of course, they hit to the point of malnutrition. In fact, we could even rescue these phenotypes by liquid gavage and force feeding these animals. As long as the researcher who would stick to the protocol, we could keep them alive as long as they were willing to give them nutrition. So this basically said that we had identified the circuit that if you shut it down, the animals overeat and become addicted to food. If you shut it off, they become aversive to food to the point of starvation. So we know this is occurring at the basal forebrain, but what else is happening? We know these neural circuits don't act in isolation. This isn't just a cell type. Where else might this basal forebrain circuitry be extending to govern these overt behaviors? So to get at this, what we tried doing and what we set out to do is basically take a genetic labeling approach where we target the basal forebrain neurons for the expression of a viral mediated reporter that's called synaptophysin GFP. It basically is a fusion protein. The synaptophysin moiety drags GFP down an axon terminal, localizes it to the axon terminal, and allows us to identify putative downstream targets. So we have a logical way to start to dissect who the basal forebrain is talking to govern these types of output behaviors. And this has been a pretty rich body of research, and we're in the thick of it right now. But so far, what we've identified, satisfyingly, first of all, we verified these outputs to the olfactory bulb. We also noted potent inputs into the lateral hypothalamus, which is known to govern feeding, into the basolateral amygdala, governing components of anxiety and aggression. And recently, we've been very interested into inputs into the lateral habenula, which is the structure that resides kind of here in the middle of the brain. And it's known to govern aspects of reward and aversion. As you can see by the synaptophysin labeling, it's robust projections to the lateral habenula. So we turned our sights onto what this might be doing as far as some of the phenotypes we're observing. However, we have a problem here because we can't simply activate or inactivate this since we have so many outputs. It's in concert, it's going to govern the activity of all of those targets. So we had to come up with a way that we could selectively dissect just the lateral habenula component. The way we did this is we turned to optogenetics, utilizing the light-gated ion channel rhodopsin, that in the presence of light, you could gate neurons to fire. We would express it in the basal forebrain neurons, but now we could systematically dissect components of neural circuitry by implanting fiber optics directly over only those inputs to gate the activity of the habenula. And for those of you who haven't seen this before, this is a typical mouse that we use in one of our models. where there's little fiber optics implanted in the different regions of the brain, and you can actually uncouple these fiber optics, these cords, and the mice live just fine. They deal with it just fine. Their behaviors are normal otherwise. They go back in their cages, and we can repeatedly bring them out and test them. So the first thing we did is fast mice overnight so they're hungry, so we could assay what this light type of stimulation to the habenula would do to feeding. We would simply put the mice back into an arena and turn the light on for 5 minutes at a time, and then measure the amount of food. Consistent with the idea of influencing feeding, every time we would turn on the light, the mice would stop eating food. If you shut off the light, they would eat food again. So this brought the habenula into this realm of feeding behavior, but we noted something also very interesting. And here again, it was a little bit of a surprise that it wasn't just feeding that we were manipulating. If we take the same animals and then establish an arena where we program a computer to photostimulate the region of the habenula every time they cross this imaginary blue line, you can see the animal retreats quickly to the other side of the cage. This is, for all practical purposes, looked to us like an escape or a massively aversion behavior, completely avoiding the area where they're stimulating this, where they're being stimulated on this side. We can document this over and over, and this is a very robust phenotype. In fact, we could change the different frequencies of light, and we could get more or less avoidance to go away from the light area that they're being stimulated. In fact, this even works when they're fasted and they're going after different types of food chow or food reward, whether it's a normal chow or a high-fat diet. You can see that they'll still they'll bolt to go get some food if they're extremely hungry, but instantly they avoid that area and go back into a point where it almost looks like an escape behavior. But remarkably, perhaps the most remarkable thing about all of this is that these animals actually never remember that they were photostimulated. Even though it looks harmful and aversive and to evoke anxiety, they seem to not even remember any of it. As soon as you shut off the light, they go to the other side without a clue or any recollection that there was an aversive cue. In fact, if we would assay these animals for fight or flight or stress response hormones, a few of which are shown here, they show no elevated innate stress. So these animals don't even recall, or they're not even aware that there's an aversive light cue. So what we've boiled down to here with this lateral habenula input is that it seems to be almost reflective. This seemingly complex behavior is a push button behavior by just stimulating these terminals to the lateral habenula, and it completely bypasses higher order processing, such as memory or even cognitive thought. And in fact, we think it, as a geneticist, we think of it as almost like the epistatic behavior that comes out of learning something aversive. You have to have a way to get there quickly. And this is just a push button trigger to aversion. We even think something like feeding and sleep might be very similar to that, or stress. There's many ways to get to stress, these hypothalamic related behaviors. So to this point, everything I've shown you has been synthetic. We've turned things off, we've turned things on, we've ablated things. We wanted to know now what's happening in the natural state. What's going on at the basal forebrain with this feeding circuitry? So to get at this, we targeted the same population of cells, once again, for a virus expressing a calcium-responsive GFP reporter known as Gcamp. In short, this Gcamp reporter, in the presence of calcium or when a neuron fires, glows brighter. So it gives us a real live, real-time readout of neuronal firing simply by looking at flashes of green cells. And so in this way, we could simply ask what's happening to the basal forebrain in real time by imaging using a microendoscope this fluorescent signal in the presence of different types of stimuli. So this is one of our first experiments, and it told us that it's worth going on. Here, this is actually a real-time movie. When we present through this microendoscope, you can see the Gcamp responses when we present food odorants to a fasted mouse. And you can see this response is very reproducible. These colors correspond to the ROIs here. And this is a very reproducible thing. Every time we present food to a hungry mouse, this is what happens. This part of their brain activates. The nice thing about this type of assay is it's genetically targetable, so it can be cell type specific, unlike electrophysiology. We can get population readouts, so we can do a number of cells at one time, and we can reproducibly and repeatedly image over a number of different sessions because the animals can live with a small microscope or a fiber optic microscope implanted into their brains. And here again, what we notice is that if you interact with food, you have an elevated response. And even if you perceive or smell food, you have an elevated response. However, this is not due to eating food. So this was kind of a clue too. This is the perception or sensory perception of food was triggering in this particular circuit. But this created a little bit of a conundrum. I mean, if you're thinking about this a little deeply, what I told you is if you shut off the circuit or ablate the cells, you become overweight and you become an insatiable appetite and you become obese. However, if you activate the circuit, you become aversive to food and you starve yourself to death. What's going on? We give these animals a food odorants, and you would think that this means that they're ready to eat. So we took advantage of kind of the high throughput nature of this and also some of our background in the olfactory system, and we did the following experiment. This is the typical imaging experiment where we do a number of different trials to different stimuli, and you can see that a subset of cells within that basal forebrain are activated by food odorants. However, if we now present the same exact animals in the same session, different types of odorants, ones that we know are associated with naturally aversive cues to rodents. So butyric acid is a metabolite that comes off of bacteria from spoiled food. And this is a known aversive cue, as well as 2-methylbutamine, which is in feces. What you can see is that this region of the brain is much, much more robustly activated, potentially overriding any natural tendency that it would have to govern food intake. However, with full disclosure, what we don't know at this point is if these are exactly the same cells or if it's a whole separate population within the basal forebrain, something we're looking at now. But as far as the circuit goes, what we learn is that this basal forebrain to lateral habenula circuit has the ability to override homeostatic feeding mechanisms. And this is kind of a key of what we're learning with this serendipitous finding associated with cholinergic basal forebrain signaling to different types of physiologically related structures. So the way we're thinking about it in summary, and this is a very broad overview, is that the cholinergic basal forebrain takes in different types of sensory cues, whether it knows what they are or what it doesn't. It assembles it, associates it with memory and experience and other types of cognitive processes to gate types of outputs, behaviors that are associated with these potent structures like the hypothalamus, amygdala, and lateral habenula. Of course, like all brain tissue, this is highly interconnected. So there's a lot of crosstalk between these sections, making it a little bit complicated to deconvolve who's the cause and the effect. But what we do know is that if you break this system right here, right at the level of the cholinergic basal forebrain outputs, you have some massive effects into all of these structures that manifest in altered feeding. What I've shown you today is feeding because it's an easy thing to measure in mice, but it manifests in altered feeding, stress, thermoregulation, anxiety, aggression, aversion, reward, and addiction. Now we can backpedal a little bit, and we've kind of put some things together since all of this, is that this shouldn't be too surprising because there's already some very nice clues that we're all aware of already out there. What I didn't talk about is all the electrophysiology we've done with respect to the signaling through acetylcholine receptors that signal through either muscarinic or nicotinic receptors. Now, even the nicotine itself should be somewhat of a tip-off here. We know that anecdotally, many people don't want to stop using nicotine-related products because it alters their urge to eat. This is also true for suppressing anxiety, and clearly nicotine is associated with addiction. So there's ample evidence that this cholinergic basal forebrain is directly involved with these potent physiological outputs. So my hope going forward is that we can identify these type of circuits, know where to intervene at specific nodes that are potent in one or another, where we can fix or target the repair of one node while leaving the other one intact. But we have to know enough about the basic circuitry in order to do that. In other words, can we take advantage of the complexity that's innately there in order to fix brain tissue? So this, coming back to this notion, and this is kind of the punchline wrap-up point, is that we all know that the brain is complex, billions of neurons, trillions of connections, etc. We also know that certain stimuli give given behaviors to everybody. We all have our own, and there's very reflexive behaviors also. We also know that if nodes are broken along the way, we have altered behaviors. This is the cause of neuropsychiatric disease. And often, I should also mention, a lot of these neuropsychiatric diseases are comorbid with many of the same things. So also tipping us off that a lot of these circuits probably share certain nodes. The idea is, can we take advantage of this complexity to somehow learn enough about the architecture to reroute information through pathways in order to correct some of these behaviors that have multiple nodes and overlapping function. The way to think about this a little simpler, and this might be pie in the sky at this point, but we're learning more and more about these overlapping circuits, The way I like to think about it is like Google Maps on your iPhone. You may have an origin and a given destination. And if you type those in, your phone spits out the most efficient pathway to that point. However, if you come across some sort of a road construction or accident, your phone can quickly reroute you to the next most optimal path. I would argue, though, that there's almost infinite numbers of ways to get to your destination, as long as you know what the roadmap is. And I think that this going forward in our laboratory, this is kind of a main agenda, is to figure out some of the main overlapping byways and freeways to come to some of these overt physiologies and behaviors that become broken in neuropsychiatric disease. Now, just to finish up, I'm give you a highlight on how we're actually trying to test this in the laboratory as proof of principle. We're taking advantage of what we know about the olfactory system, and we've implemented this new kind of high throughput odor stimulation device that has this turret of different odorants that we can connect onto a delivery system. At the same time, we implant animals at given circuit nodes. In our case, we're interested in these feeding circuits because they're very easy to measure for the expression of Gcamp. And we've teamed up with Dr. Damian Young here at the NRI at Baylor College of Medicine, who is a pharmacologist and chemical biologist, and he's made a library for us that's based off of either piperazine or nicotine-like backbones that we can change functional groups to change the binding properties of them to the olfactory-related receptors in the olfactory system to gate sensory perception of different types of food or food-blocking cues. In this way, what we hope to do is image the downstream circuit nodes that we think are directly involved with something like appetite. Appetite is just one behavior, but for us in the laboratory, it's very easy to measure, where we can see differential responses for compounds to either invoke feeding or to invoke feeding cessation in a way that by simply augmenting or pirating the pathway from the olfactory system, we can govern appetite. And in fact, this has become a reality. This is an image. You can't really see the mouse is sitting back here, but it's sitting here awake, doing fine on a treadmill, and this is his nose right here. Here's the odor gun and the Gcamp imaging system. We do this multiple ways, either with the microendoscopes or with 2-photon microscopes. And we monitor other things like respiration and pupil responses, because we also know that the cholinergic signaling pathway is associated with arousal, motivation, reward, etc. So this is one typical way that we're trying to get at this idea of, can we co-opt intact circuits to change physiologies and behavior in hopes of utilizing these type of approaches in the future in other parts of the brain. So with that, I'm going to wrap up and definitely say my thanks. First of all, I want to give my heartfelt gratitude to the Edith and Peter O'Donnell Foundation for their generous support of this whole mechanism. This is a fantastic thing we have in Texas, and I'm amazed by the community. Of course, TAMEST for their effort in putting all of this together, and every year recognizing efforts going on, not only in the scientific level, but the mentorship and collaborative levels too. So I appreciate all of the effort they do towards keeping this mechanism going forward. I also, the biggest thanks to my laboratory. I have an absolutely wonderful laboratory. This is a little bit deceiving because this was a picture from last year or so, back when we could be a little bit more dignified and we could take a picture as a group. This is kind of what we look like now at a week-to-week basis when we collect as a group. But I must say they've all been highly motivated and weathered the storm very well. I have a fantastic group, and they push this work forward and make me excited every single day. So I thank them with all of my heart. The collaborators on the work is Quinchun Tong from UTH. He's been our brain behind the feeding projects. This is his expertise, and he's an absolutely wonderful collaborator. The alumni that actually spawned this work, Isabella and Alex Herman, who discovered the original phenotype from the beginning. Of course, generous support from many different institutes. My institution at Baylor College of Medicine, and of course the Jen and Dan Duncan Neurological Research Center, which we have a great local community that supports us here, as well as the directorship of Huda Zogby, which is just a fantastic environment. And then finally, I want to acknowledge the late Bob McNair, who he and Janice, through the McNair Foundation, recruited me here originally to start my laboratory from the first place through a generous startup package, and really highlight the type of philanthropy and support we have as scientists here in Texas as just very good human beings. And I'm so appreciative to be in such a great environment. And in fact, this is just an endearing comment that I remember him from, that I like to I think I like to live by that too. So with that, I appreciate all of your attention. Even though this is Zoom, it's always weird. I would be happy to take any questions. So thank you.
Well, thank you very much. That was an extremely exciting talk. And as we've said, I think the potential for new interventions is clear. So could I just ask one question? What do you see as the mechanism for showing that these kinds of complex behaviors and the mechanisms for them also apply to primates, including us? For example, do you think there's any way you could use functional MRI to see what happens with different cultures, people from different cultures who are exposed to fermentation smells that are objectionable to some cultures and pleasant to others? Would they show the kinds of differences you'd predict from your animal studies?
I think that's an absolutely great question, because there's two sides to that coin. First of all, there's the innate kind of homeostatic and sensory processing cues that we can't override. So for example, you might be very hungry right now, and if you were blindfolded and you smell a disgusting odor, and we'll get to the cultural component of that, but if you were to smell something that's naturally aversive to you, it will completely suppress your appetite. Likewise, you might be already satisfied or sated with a meal and you smell something extremely savory, or you might be hungry, and if you smell something that's appetizing, it will get your gut rumbling and you will have hunger pains just because the anticipation of what's coming. So that's kind of the hardwired component of that. We talk about this idea of cultural selection of what's good sensation and bad sensation all the time. And in fact, we play this trick on mice all the time in the laboratory. So we condition them to different odorants to either be good or bad odorants related to a reward like food or water or not, even if they're naturally aversive. So you can override, you can build homeostatic mechanisms. I think the homeostatic mechanisms are cultural, learned, and based on experience and sensory past. And I think that they can be overridden easily. You can rewrite that memory bank based on your experience level. With that being said, I actually think it's well worth going into the studies in humans to ask exactly those questions. What are the differences? What circuit nodes are being implemented here or there? Clearly, the stuff I didn't talk about at all is that memory, experience, and these other parts of the brain are directly impinging upon the circuit node in the first place. So it's a great question. I think that there's a lot of room for trying to dissociate in humans the differences between how we perceive something as being good or bad. And culturally, the values are going to establish all of that and what we've learned. It'll remain to be seen. But I think ultimately, all of these homeostatic mechanisms ultimately push button these reflexes, whether you're going to be hungry, you're going to be stressed out, or not. So I think the real potential here is to be able to circumvent the non-homeostatic mechanisms. So I think those are the ones we have to try to override.
Thank you very much. So we have a question from Paul Faffinger. How would you think about it if the same cholinergic neurons are regulating all these different behaviors? Does this mean these behaviors are always linked, or that the cholinergic neuron responses are differentially impacting the different areas, either by differential synaptic release, presynaptic inhibition, or what?
Great question, Paul. That's I think both. I think that I have a pet hypothesis that the cholinergic neurons, they're so robustly innervating different sensory areas, and the different sensory areas are comprised of completely different cell types. For example, the olfactory bulb is heavily populated by interneurons. So that area is largely going to be a lot quieter than the cortex that's heavily populated by excitatory neurons. So I think there's going to be local release properties that are going to gate this. But I also think that along the way, the other parts of the brain are going to short circuit release properties here and there to govern at the cell biological level. An interesting side note is that conditioning, motivation, and reward and arousal, if you anesthetize mice, for example, none of these type of, the basal forebrain, if you anesthetize a mouse so they're not aware of what's going on, there's no consciousness to them, the basal forebrain doesn't work. So it's a good question experimentally of whether or not you can train animals to think of something as being rewarding or aversive without the basal forebrain working at all. I don't think acetylcholine is going to act the same way in every area. long and the short of it. I think that there's going to be local release properties, and I think there's different intersectional nodes where it's going to work differentially. And finally, real quickly, Paul, is that the other population of cells that I didn't have time to talk about at all is that it's not all just the cholinergic cells. In the basal forebrain, the cholinergic cells have enslaved projection neurons differentially, both glutamatergic and GABAergic projection neurons that have differential outputs. So a lot is happening locally that have output channels that are discrete.
Thank you. And the final question is from Anne Solomon. I apologize if I pronounced your name incorrectly. How did you select the specific chemicals to test in the final series that you presented?
Oh, yes. So good question. We actually didn't select them. We handed that over to our collaborator, who's the biochemist, who basically, this piperazine backbone, to my understanding, is kind of a common building block for biological compounds. And importantly for him as a synthetic chemist, he's able to couple different functional groups onto that backbone to have completely different binding properties to different biological receptor subtypes. So we actually went into it not really wanting to know the logic behind it. We wanted to do an objective screen, and we're just initiating that screen right now. And we're excited because we are having differential responses right off the bat between different backbones. So we're at iteration 1. We have the backbones, and we're figuring out which ones have differential responses. And then we're going to go to the next layer and deal with different functional groups to see if we can get selectivity.
Great, thank you very much. Well, I think that brings us to the end of our session. So I'd like to thank you again for a superb talk, very stimulating in many different ways. And hand back to Terence for the farewell.
Thank you very much.
Thanks to both of you, and thanks to all of you for joining us today. We're really happy to present these sessions, and hope that all of you will join us for our future sessions. We're going to have one in 2 weeks at the same time on Wednesday, February 10th, for our recipient presentation from Dr. Guiha Yu from the University of Texas at Austin. He's the 2021 recipient of the O'Donnell Award in Engineering. And TAMEST member Joan Brennicky from UT Austin will moderate that session. We're going to have the video and slides from today's session up on our website tomorrow. And all of you that attended today will also receive an email with links to that video and those slides. You'll also get a survey at the end of this session for your feedback. We'd love to hear it. And for those of you that are interested in nominating a candidate for the 2022 O'Donnell Awards, we're now accepting applications, and you can find information about the nomination process on our website, which is tamest.org. We'll also have a link to the awards nomination page included in that follow-up email that we'll send you tomorrow. So we encourage you to review that information and help nominate the next class of Rising Star researchers in Texas. Nominations will close on April 30th. So that concludes our session for today. Again, thank you to our moderator and to Dr. Arenkiel for presenting your research. Congratulations on your award. We hope to see as many of you as we can in 2 weeks for our next session. And please stay safe and be well. Thank you again.